What is genetic engineering? Genetic engineering is a process of altering an organism's genetic code to produce a desired trait. This is done by taking the gene of an organism and transferring it into another organism, such that the gene is expressed in its new host. This is allowed for the production of transgenic organism, which is one that carries DNA sequences that it would not normally have. The production of transgenic organisms has raised many social and ethical issues. Despite these challenges, genetic engineering has greatly impacted some fields such as medicine and agriculture. Some applications include the production of drugs and improving resistance of plants. Before we look at genetic engineering, we need to know how DNA works. DNA is the basic code of life found in every living organism. And this DNA is made up of genes, which in turn are made up of nucleotides consisting of 4 different bases A, T, C, and G. Certain genes in the DNA code for specific proteins, which then code for the different traits of an organism. Scientists can then edit the DNA to alter the traits of an organism. Scientists cut DNA using a pair of molecular scissors called restriction enzymes. Restriction enzymes recognize 4 to 8 base pair sequences called restriction sites and cleave both DNA strands at its site. Some restriction enzymes may cut both of the DNA strands in the same place, which will produce blunt ends, or they may cut the DNA strands in different places, leaving uneven DNA sequences called sticky ends. For now, we will look at sticky ends only for genetic engineering. These sticky ends can bind to another strand of DNA with sticky ends through complementary base pairing. This produces a recombinant DNA. When the DNA strands are joined, there are gaps in the sugar phosphate backbones of the DNA strands, called nicks. Each nick is sealed by an addition of a phosphate group by the enzyme DNA ligase. A vector is a carrier DNA molecule into which a DNA fragment containing the wanted gene can be inserted. The result is a molecule of recombinant DNA. Many different vectors are used in genetic engineering. The most commonly used vector is a bacterial plasmid, which is a small circular DNA molecule. The key features of cloning plasmid include the origin of replication, selection markers such as antibiotic resistance genes, and multiple cloning sites to facilitate the insertion of gene of interest. Now, we look at the process of genetic engineering. The first step is isolation, followed by ligation, transformation, selection, cloning, and lastly, insertion. To obtain the gene of interest, the gene has to be isolated from the entire genome. First, restriction enzymes are used to cut the DNA of the genome into fragments. The fragment containing the gene of interest is then purified to get rid of other undesired parts. A bacterial plasmid containing a LEX-Z gene and an ampicillin-resistant gene is cut with the same restriction enzyme to produce complementary sticky ends. The restriction site is found within the LEX-Z gene. The fragmented gene of interest and fragmented plasmids are then mixed together and ligated with DNA ligase. The sticky ends may join up in 2 different ways, giving unaltered plasmids without the gene of interest or recombinant plasmids containing the gene of interest. Since the plasmid has its restriction site within the LEX-Z gene, this means that inserting the gene of interest into the plasmid renders the LEX-Z gene non-functional. During transformation, competent cells are prepared by treating bacterial cells with divalent cation ions. The plasmids are then introduced into the competent bacterial cells through heat shock transformation, resulting in 3 possible outcomes: bacterial cells that did not take up any plasmid, bacterial cells that took up unaltered plasmids, and bacterial cells that took up recombinant plasmid. In selection, the transformed bacterial cells are cultured on agar medium containing ampicillin and agar and incubated overnight. Bacterial cells which did not take up any plasmids lack the ampicillin resistance gene and are not able to survive in the agar medium, whereas bacterial cells with unaltered or recombinant plasmids are able to grow on the agar medium. Functional LEX-Z gene on unaltered plasmids produce an enzyme that converts agar into a blue product. As a result, bacterial colonies containing unaltered plasmids appear blue, whereas those containing recombinant plasmids appear white. Colonies containing recombinant plasmids carrying the gene of interest can then be grown in large cultures, allowing the cells to multiply rapidly. Other methods of inserting a foreign DNA into a cell include microinjection, where a glass micropipette is used to inject the DNA, and electroporation, in which electrical pulses produce pores in the cell membrane for DNA to be taken up. Genetic engineering has opened up many astounding possibilities for transformation of plants and animals, and even humans. Genetic engineering might allow for an evolution of intelligent species. It might end diseases. It might even extend our life expectancy by centuries. No one knows what our future will be like, but it is approaching. Thank you.